Okay, we're back. Um, so we stopped here. Table sugar. Table sugar is sucrose. So they must be broken down into monosaccharides so that they could be used for energy by organisms. This is why usually if you need a lot of energy, you eat a lot of carbohydrates because this is the chemistry behind that. So yeah, disaccharides, double sugar, two monosaccharides are joined by a process called glycosidic linkage. So this is an important term. Okay, lactose intolerance. So what is lactose intolerance? It is a condition in humans who lack lactase. This is an enzyme that breaks down lactose. So as a result, you avoid eating dairy because dairy has lactose. So you don't have the enzyme to break down dairy, the lactose that is inside. Well, dairy has lactose, so you can't basically um, break it down. So yeah, uh, it's similar to the idea that we cannot eat grass. Although grass is nutritious for cows, um, like yeah, we don't have the enzyme uh, in order to digest grass in the same way as them. I know there's a specific enzyme for that, but I think we might see that in a different card. We'll see. But at least I gave you a heads up already. Okay. This is where mammals stock their long term food reserves, which become fats. Adipose cells. Adipose tissue, on the other hand, it cushions vital organs, and the fat layer serves as body insulation. This is what penguins and animals that live in temperate and Arctic climates use so that they keep themselves warm. Polar bears, among other animals. So, yeah. So, what are one of the organs that it cushions? One of them is the kidneys. Okay? Next. I'll just fix this for a while. I know that you can already see the answers here, but still try challenging yourself. Okay. Polysaccharides. These are macromolecules and polymers with a few 100,000 monosaccharides joined together by, again, a process called glycosidic linkages. So, we have two types of polysaccharides, type 1, type 2. So, for type 1, we have storage polysaccharides. They serve as storage material. Like, literally, from the word storage, they store. They are hydrolyzed as needed to provide monosaccharides for the cells. So one example is starch. Starch is a polymer of glucose monomers. They are stored by plants as plastids, basically granules with cellular structures, and they are withdrawn by plants by hydrolysis. The simplest form is amylose. A complex form is amylopectin, and animals store glycogen. Most animals and humans have enzymes that are able to hydrolyze plant starch, making glucose available for cell nutrition. I think technically grass is considered a plant. So, but even so, humans cannot break down the what do you call this? They can't break down uh, I'll continue what I've started saying just now later. I just can't remember the word. But basically, yeah, humans cannot hydrolyze this the simpler substances found in grass. You just can't. Okay? I think it's cellulose. Yeah. Humans cannot digest cellulose because we don't have cellulase. Cows do. Okay, that's it. Type 2, we have structural polysaccharides. They serve as building material for structures that protect a cell or a full organism. So cellulose is a major component for tough plant cell walls, and it is the most abundant organic compound on Earth. Chitin. This is embedded in proteins. It forms an exoskeleton of arthropods and hydrolyzed. They undergo hydrolysis. This is the reverse process of dehydration reaction, or basically adding a water molecule to turn polymers into unchained monomers. So this is in my own words, from how I understood the syllabus's definitions. Okay. Steroids. Lipids with 4 fused rings in its carbon skeleton. So again, like what I said earlier, 3 types of lipids under it. We have fats, phospholipids, and steroids. Steroids is number 3, the third type. So kinds are distinguished by the specific chemical groups in the rings. We have cholesterol. Cholesterol is under steroids. This is a component in animal membranes. It is a precursor for the synthesis of other steroids, and for vertebrates, this is made in the liver. And yeah, lipids such as sex hormones, estradiol or estradiol, testosterone, etc. Yeah, I think these are synthesized by cholesterol. I think that's what it means based on what's stated here. We'll clarify this more later on. Okay, lipids. So we have fats. It's the first type. In other words, it's called a triglyceride or a triacylglycerol. Triacylglycerol. That's a long word. It just basically means fats. So, what is the reason why fats separate from water? Why is it hydrophobic in the first place? Water molecules hydrogen bond to each other, and they exclude fats in the process. Why are fats hydrophobic? Relatively nonpolar CH bonds in the hydrocarbon chains of fatty acids. It's not a complete sentence. Anyways, I cannot make a full explanation on that anymore. It's been a long time since I've made this deck. So, in making a fat, each fatty acid molecule is joined to glycerol by dehydration reaction, causing ester linkage. So, this is a bond between hydroxyl and carboxyl group. What's the major function? Energy storage in adipose cells or tissue. So again, dehydration reaction is responsible for creating polymer chains. Glycosidic linkage. Glycosidic linkage is for carbohydrates. For lipids, it's particularly for fats. It's called ester linkage. Hydroxyl carboxyl group. What's the major function? So energy storage in adipose cells or tissue. And I believe the reason why fats are excluded in hydrogen bonding of water is because water is polar and fats, like oil, are non-polar molecules. So polar and non-polar molecules cannot mix together. So it explains these two statements here, enforces the idea that is being expressed there. Okay? What is the diffusion of free water molecules across a selectively permeable membrane? Osmosis. Still remember it. Nice. Okay. To where there is fewer solute, there is more free water molecules. So I believe solute means, for example, salt. So there is more water if there's less salt. I think that's the idea. In the other side, um, where there is more solute, there are less free water molecules. How do I explain this in a better way? Um, so the tendency is where there is more solute, that's where water goes. So here you can see that there's a higher water level here on the right side. That's because there's more solute in this side. So the tendency is water would go there. So water will move to the right, solutes will move to the right. Also solutes. Okay, more on this later. I believe this is not the only flashcard about osmosis. Okay, what is the bond between hydroxyl group and carbonyl group? So this is the result of the formation of fat by glycerol plus 3 fatty acids by dehydration reaction. Ester linkage. So if glycosidic linkage again corresponds to carbohydrates, ester linkage corresponds to fat, formation of fat. And we have monosaccharides. Generally, they have molecular formulas with CH2O. Glucose is the most common monosaccharide, basically sugar. It is important in life chemistry. It includes monosaccharide trademarks. So one is carbonyl group, two hydroxyl groups, and we have aldose and hexose. What is the criteria for classifying monosaccharides? So we have three, I think. First is the location of the carbonyl group, aldose or ketose. Let's see if there's a visualization for that. Okay. So either aldose or ketose. Aldoses or aldehyde sugars, basically the carbonyl group is located at the end of the skeleton. Where do we see that here? For ketoses, on the other hand, or ketone sugars, the carbonyl group is within the carbon skeleton. Okay, second criterion, the size of the carbon skeleton. Usually it's 3 to 7 carbons long. And number 3, the spatial arrangement around asymmetric carbons. So for aldose, aldehyde sugar, an example is glucose. For ketone sugars, one example is fructose. Where do we see that here? Okay. I believe for the first criterion, basically for aldehyde sugars, such as glucose, the CH is at the end. Where is that? The carbonyl group. Carbonyl group is at the end of the entire skeleton. For ketone sugars, the carbonyl group is within the skeleton. It's not in the edge. So as you can see, it's technically still inside the skeleton. Okay. Triose is a 3-carbon sugar. Pentose from the prefix pent, 5. Pentagon, 5 sides. Pentose, 5-carbon sugar. Lipids are a diverse group of hydrophobic molecules. It does not include true polymers. Okay, this has been mentioned earlier already. Molecular logic of life. Small molecules common to all organisms act as building blocks that are ordered into unique macromolecules. So, despite immense diversity, molecular structure and function can still be grouped roughly by class. So, there are four major classes of biological molecules. Okay, key concepts. Okay, let's focus here first. So, macromolecules are polymers. They are built from monomers. Carbohydrates serve as fuel and building material. Lipids are a diverse group of hydrophobic molecules. Proteins include a diversity of structures, resulting in a wide range of functions. One of them, I believe, is making your muscles, building the structure of your muscles, among a lot of other things. I remember making this list of protein functions, its relevance in making the physical structure of your body, not just in terms of muscles. I'll see if it's also included in this deck later. We also have nucleic acids. They basically store and transmit and help express hereditary information. So anything genetic that is, we refer it to nucleic acids. Genomics and proteomics have transformed biological inquiry and application. So what is genomics? Basically studies the entire set of genes in a genome. On the contrary, for proteomics, they study basically studies the entire set of proteins produced by a cell. Two types in genomics: structural and functional. Genomics involves sequencing, analysis, and mapping of genomes. For proteomics, there are three types: functional, structural, and expression proteomics. Okay. I don't think this is going to be that important. I don't remember this being in the grade 12 exam. But at least this is just for extra information. Disaccharides. Okay, this has been discussed already. Next. Okay, discussed. Okay, new card. 5 and 6 carbon sugars form rings because they are most stable. They are the most stable form of these sugars under physiological conditions. Chemical equilibrium between the linear and ring structures attracts the formation of rings. Frick, I'm not sure if I can still explain what the illustrations mean anymore. Next, carbohydrates serve as fuel and building material. They include sugars and polymers of sugars. They are the simplest carbohydrates, monosaccharides or simple sugars. They are monomers from which more complex carbohydrates are built. So we have sugar or saccharose, honey is fructose, milk is galactose, maize or basically corn is mannose, apple is glucose. Dehydration reaction versus hydrolysis. So for dehydration reaction, this is the synthesis of a polymer. Hydrolysis is the breaking down of a polymer. Okay, now we have an illustration on the two processes. So for dehydration reaction, again we are synthesizing a polymer. and this is at the expense of losing a water molecule. So as you can see here, water is expelled. But in hydrolysis, on the contrary, the breaking down of a polymer, so hydrolysis adds a water molecule, and from there it causes the breaking of a bond. Okay. Monosaccharides, or glucose specifically, are major nutrients for cells. In other words, they fuel cellular work. One example is cellular respiration. So that's one kind of cellular work. It is very significant in maintaining life in the most basic unit, that is the cell. Okay, its carbon skeletons are raw things that can make other organic molecule types like amino fatty acids and fatty acids. Monosaccharides that are not used in the said ways eventually become monomers in disaccharides or polysaccharides. So in cellular respiration, this is where cells extract energy from glucose molecules by breaking them down in a series of reactions. So this is what happens every time you consume carbohydrates. Okay. Factors to consider to observe cell behavior in a solution. Solute concentration. Okay. To observe the behavior of a cell in a solution, we consider solute concentration and membrane permeability. And both are taken into account for tonicity. What is tonicity? This is the ability of a surrounding solution to cause a cell to gain or lose water. Okay, that's basically what was stated here. Gotta fix this. Okay, so we have two kinds of fats. We have the healthy type and the unhealthy type. For this is the unhealthy type, the saturated. For unsaturated, this is the healthy type. Olive oil, salmon, stuff like that. For saturated, it basically also has bad cholesterol, fast food, etc. Okay, so for saturated fatty acids, they are solid at room temperature. I'll break this to you now. Because they are solid at room temperature, saturated fatty acids usually it is responsible for causing the buildup of plaque in the blood vessels, particularly the arteries, and this is why we have a disease called atherosclerosis. Blood flow coming from the heart to the rest of the body is not achieved properly because in a way there is a blockage in the arteries because of solid fatty acids, in other words saturated types. So in other words, what describes saturated fatty acids? There are no double bonds between carbon atoms, so the structure is saturated with hydrogen. One example is stearic acid, and it makes saturated fat. There's also an illustration here. We'll get to that later. So for unsaturated fatty acids, they are liquid at room temperature. It doesn't cause any blockage. It doesn't cause any atherosclerosis. So there is one or more double bonds with one fewer hydrogen atom on each double bond carbon. So basically less hydrogens. There are several types that make unsaturated fat. One example is oleic acid. Okay. Levels of triglycerides or fats. Okay. So for saturated, this is what it looks like in simple illustration. There are no double bonds between C atoms. So one line. So in a single carbon atom, there is a maximum of 4 hydrogens that can attach to it. So this is what it looks like. The structure is saturated with hydrogen. On the contrary, for unsaturated fatty acids, there's a double bond as you can see here. So with one fewer hydrogen atom on each, 2 times bond C. Okay. Next. Okay, we've already discussed this. Also this. Double sugars, disaccharides. There you go. Double sugars, basically they consist of 2 monosaccharides joined by a covalent bond. For polysaccharides, it's the same thing, but more than just 2 monosaccharides. The union of 2 monosaccharides. Carbohydrate macromolecules that are polymers, they are composed of many sugar building blocks. This is a lot of information. We'll just move on. Existence of cells depends on the properties of phospholipids. Yeah. No further explanation for that. So what are phospholipids? Okay, so this is the second type of lipids. The second type, yeah. So they are essential for cells. Phospholipids are major constituents for cell membranes. This is what I was talking about earlier. The phospholipid bilayer. Basically the cell membrane. So in the making of a phospholipid, we just add a glycerol with 2 fatty acids. That's it. The 2 ends of a phospholipid, we have the hydrocarbon tail, which is nonpolar, and the phosphate group head, it's polar. I hope there is an illustration. It appears to be that there is no illustration here. But basically, these 2 ends form a phospholipid bilayer, and it forms a boundary between the cell and the external environment. They establish separate compartments within eukaryotic cells. So there's a reason why it's important. Well, it is important to know that the hydrocarbon tail is nonpolar and the group head phosphate is polar. More on that later. Okay. It appears to be that this is also important to know. I did not add this in Anki, so it's already here in the picture anyways. So glycerol, its third hydroxyl group is joined to a phosphate group. I think these are just examples. Okay, we've seen this earlier. Okay, the tonicity of a solution depends in parts of its concentration in solutes that cannot cross the membrane relative to that inside of the cell. Okay, so for tonicity, we have in this table animal and plant cell. We're comparing the two in 3 kinds of environments. In a hypotonic environment, an animal cell, we all know that plant cells have cell walls, animal cells do not. So animal cells, there's a tendency for animal cells to get lysed or for them to burst, because in a hypotonic environment there is little solute and there's too much water. Too much water enters the cell, because there's no cell wall to enforce it from um, the surge of water coming from the external environment. So yeah, the tendency is um, animal cells get lysed or they burst. Plant cells, on the other hand, are turgid in this kind of environment because there is a lot of water outside, but because there is a cell wall, it is protected properly. It the cell wall basically keeps them from getting lysed. So I believe this is the most ideal environment for a plant. Maybe not all, but um, looking at the illustration, the best shape of a plant cell is turgid, if I remember correctly, from my teacher. Okay, hypertonic environment. This is the other extreme. This time there's too much solute. Uh, there is too little water. So the tendency for an animal cell is it gets shriveled. For a plant cell, um, the plant cell isn't exactly dead if there's not much water outside, um, but it becomes plasmolized. And this is not, well, it is not good for the plant. Um, its stage of being in plasmolysis would only last for a long time. It wouldn't last for a long time. It's just like saying you haven't watered your plants for a long time. Eventually it would wither. That's the thing. Okay, so the environment that well, say that it's the normal environment. You call it isotonic. So for the animal cell, it's normal. So there is a balance in the proportion of solute to solvent, salt to water. In the plant cell, um, of course, it's better for its shape to be turgid, but it's not exactly that bad for it to be flaccid. Um, so yeah, I believe this is the manifestation that you see in plants that do not live underwater. So yeah, the external environment, sometimes it rains, sometimes it's sunny. So yeah, it's flaccid as a result. Okay, this has already been mentioned. We're finished there. Okay, we haven't seen this card yet. Macromolecules. Macromolecules are large carbohydrates, proteins, and nucleic acids. They consist of huge chain-like molecules called polymers. I know this flashcard is a little repetitive already. We've already mentioned these terms earlier, but Anki is for memory retention after all. Better to be repetitive so that you remember. Okay? Polymer is a long molecule consisting of many identical building blocks linked by covalent bonds. For a monomer, there are smaller molecules, repeating units that serve as building blocks of polymer. Okay, mono is one, poly is many. Okay. Hydrogenating vegetable oils makes saturated fats. Okay, so it was mentioned earlier that in saturated fats, there's only a single bond. Therefore, there is a lot of hydrogen molecules. So, the more hydrogen, basically, the more solid the structure is. That is for saturated fats because, again, they are solid. So, vegetable oil initially is liquid. It doesn't. It's not. It's not bad. It doesn't cause atherosclerosis unless it undergoes the process of hydrogenation, basically you add more hydrogen molecules, so it wouldn't be any different from saturated fats, because saturated fats again has a lot of hydrogen molecules. So it causes saturated fats okay. um, I'll scrap what I just said. I would no longer continue what I said. Um, so eventually they become trans fats. So recall that hydrogenation is a process of converting unsaturated fats like vegetable oil. Initially, vegetable oil is healthy. You turn it into solid form, saturated fats, or in other words called unsaturated trans fat. So trans fat, we all know that it's associated with the idea of not being unhealthy for the body. So unsaturated fats are liquid form at room temperature. Saturated fats are solid form at room temperature. Just to recall what was stated earlier. We hydrogenate to make naturally liquid fats in solid form, such as margarine and butter. So these are trans fats. Okay, polysaccharides. Just kidding. Macromolecules. Okay, we have finished the deck. That's all for this video. Thanks for watching.